the relationship between this mitosis and meiosis is when you look at the cell, okay, you need to observe about the chromosomes, the number of the chromosome inside. So for example, right, okay, let me show you one picture, okay. This picture is very nice. This is actually a textbook for the DSC's biology, okay. It's not under IGCSE, but I just want to show you some of the picture. It's quite interesting to know. Yeah, so you can see, right? So for example, you can have a look about this. Okay, so this picture showing about what? This picture showing about the mitosis. So actually, what is mitosis? Mitosis is referred to the, they refer to the chromosomes number is undergo replications, you know, which is the DNA after they undergo replication. So the DNA from one copy, right, they will, after they undergo replication, they become 2 copy like this. So they will have like this, okay. So you can see, right, this is a chromosome. So chromosomes are in the actual image, right, they just look like this, look something like this, okay. So this is a chromosome. So and then you should note the difference, okay, the difference in the chromatins and the chromosomes. So you can see this is a chromatins. Chromatin, right, is like something like a thread-like structure. Just look at the thread. You can see, right, inside the threads inside this nucleus. So actually this is a chromatins. Okay, so you can see, right, the chromatins. So the chromatin is actually produced from the DNA. Means the DNA need to combine with the histone proteins. So DNA plus the histone proteins, they will become the chromatins, which is the long thin fibers. They just coil like in this way. So after that, the chromatins, right, they will undergo the condensations. You know, they'll undergo condensations. So which is the chromatins, they will condense. So when they condense, right, or they will coil to each other, because chromatin is just like thread. So when the thread, they just coil to each other, right, they will become the shorter and thicker form, which is this one. So this is a chromosome. So at this way, you can have a look. Yeah, this is a chromosome structure. I just look like this in actual way. Yeah. So when we talk about chromosomes, it's referred to something like this, okay? Like something like requires what? Butterfly-like shape, yeah. So after that, they will do the requires the cell division. Okay, so this is called what? Nuclear divisions. Because mitosis, the first step is what? The first thing is they will do the DNA replication, right? So you can see this is the first step. They will do this DNA replications. What's mean by DNA replication? It refers to they will make another copy of the DNA. Something like from one copy, right? They will make another photocopy, become like this. So like this, 2 copy, right? 2 copy, just showing like this in this way, 2 copy. So this is a chromosome, right? So actually inside showing about 4 chromosomes. So this is the 4 chromosome. So every 4 chromosomes, they will have about 2 sister chromatics. Because what's meant by sister chromatic? Sister chromatic is referred to the request what? The chromatin fibers after they undergo the DNA replications. So they will have like this, okay, sister chromatic. So you can have this one, you can see prophase, yeah, metaphase. So which is the chromosome, right? Initially, they will condense like this way. So this is like the preparation stage. Before they do the cell division, they need to prepare first, right? So how they prepare is they will condense and coil it become like this, which is this one is a chromosome. So after that, the chromosome, right, they will just align at the central part. So you can see, right, this is a chromosome. They just align in the central part like this. So this is what? This requires the metaphase. So you can see chromosome, they just line up in the middle of the cell. Yeah, so this requires what? Equator. Yeah, so with the spindle fiber, so you can see, right, the spindle fiber, they just attach to the central part of the chromosome. See, central part, which is they refer to this one. Okay, you can see, right, between it, there will be the like central part. Actually, this is called what? Centromere. Okay, centromere. And then they will proceed to the anaphase. So when they proceed to the anaphase, right, what will happen is the chromosome just spill. They separate. So you can see, right, this image showing what? Showing the chromosomes are being separate. due to the shortening of the spindle fiber. So that will be the spindle fiber. The spindle fiber, they just attach to the chromosome, and then they'll split them up. Like this. Okay, so spindle fiber contract, so they will separate the sister chromatids. So after the separations, right, the sister chromatids, now they will become chromosomes. Yeah, they will become chromosomes. And then they will just enter to the telophase. So you can see, and then you can see they just reform by the nuclear membranes. And then the chromosome, right, the chromosomes, now they will uncoil. So you can see, right, chromosome, they just uncoil, become the long thread-like structure, which is a chromatin again. So this is the process happens in the mitosis, in the stage of the nuclear divisions. So this is how they will divide the nucleus from one to become two copy. So how they can do this is, first they need to do the DNA replication first, because they need to duplicate the chromosomes found inside the nucleus, so they can able to divide them. And then followed by this, it will be what? It will be the cytoplasmic divisions. Okay, you don't need to know about this term. It's not your level to know. Okay, you just need to know about, first is DNA replication, followed by the nuclear divisions, followed by the cytoplasmic divisions. So you can get a two daughter cell, which is genetically identical to the parent cell. So this is the process that you're able to get. So both is same for the animal cell and the plant cell. Okay, so this is mitosis. Let me get back to you about the lecture. Yeah, so you can see this whole process is called as what? Mitosis. So as you can see, right, DNA, they just undergo replication. Just like in this way. Okay, initially the chromosome, right, is just like this. Yeah, let me show you. This is inside the nucleus, right? They will have the chromosomes. So the just chromosome, they just like this. Like this. Okay, just one copy, one copy. And then here will be another, like this. So like this is we call as what? We call as chromosomes. This is we call as chromosomes. So after that, first they need to undergo DNA replications. So after they undergo DNA replication, right, they will become like this. Now they become like this. Okay. So each of the chromosome, right, they have their another copy. We call as what? Sister chromatid. So at this way, you can see like this, right? They will have another copy. Now they become like this, produce another copy, because DNA is being replicated. Which is you have another copy. You have another copy of the DNA. Okay, another one will be like this. This. So you can see each chromosome is being duplicated, and then you will continue to the phase. Okay, actually you don't need to know the phase. And that's why you don't need to know about the phase, which is what the prophase, metaphase, anaphase. You are not required to know for the IGCSE syllabus, but you need to know the 3 steps: DNA replications, nuclear divisions, then followed by what? Cytoplasmic division, which is the term known as cytokinesis. Cytokinesis. This is not really come out in the exam, but it is very important for you to know. But maybe it will come out in exam, I'm not sure. Right? So that's why after they line up at the equator, and then they will just divide by the shortening of the spindle fiber. So now they will become like this. So you can see, right? Yeah, each of the cell, okay, each of the daughter cell contain the chromosomes. So these 2 daughter cell, okay, and that's why these 2 daughter cell will them have a same genetic material like in the parent cell? Yes or no? These 2 daughter cell, do they contain the totally same genetic material like in parent cell? Yes or no?
Yes.
Yes, correct. Okay. The daughter cell, right? Yeah, the daughter cell, okay, which is they produce in the mitosis, is what is genetically the term genetically identical to the parent cell, to the parent cell. So this is mitosis. Oh, where is it? So this is mitosis. Okay. Then how about the meiosis? How about the meiosis? Where do I go? Okay. Okay. The meiosis, you can see, right? The meiosis. Okay. Let me show the actual picture for the meiosis. Meiosis, right? Okay, you can refer to this. This I'll send to you, okay, for this book. I think it's very good for your understanding. Okay, so let me show you about meiosis. How is the process? So actually meiosis, right? When we talk about meiosis, meiosis is due to what? It's a process. It's also considered as a cell divisions, but the meiosis, right, is mainly they want to produce the 6 gamete cell, which is the sperm and the ovum. Okay, in the plant, it will be the pollen grains. Yeah, so this 3 cell is product of the meiosis. So why they need to produce meiosis is because that you can see there will be the sperm and the ovum. Each of the gamete cell, they will have about 23 number of the chromosome. So in order to restore the diploid number of chromosome, right, they need to half them. I mean, they need to divide the chromosomes into, into half number. Yeah, so that's why sperm, they have 23, ovum, they'll have 23. So when they combine together, they'll become 46. So this 46 chromosomes is the human chromosome number. Because in the normal human chromosomes number, right, it should be about 46, not more than that or not less than that. Okay, it cannot be 45 or it cannot be 47. It will be disaster, okay? It will be something like genetic disease if they have this kind of like wrong in their chromosome number. Because if they produce a cell, if they produce one cell, okay, which is this one, zygote. So that's why zygote is very important. It's because actually the first cell they produce is zygote. Yeah, so Ethan and Suyang, both of you are also produced from zygote, which is a diploid zygote. After the sperm and the ovum, they just fuse together by the fertilizations, they'll produce diploid zygotes. So these diploid zygotes, they contain about 46 chromosomes, and that one diploid zygote, right, they constantly do what? So now they will do the mitosis. They will do the cell divisions, which is this. Okay, they will divide. They will divide, become the ball of cell, become the morula and the embryo, and then now they will become the baby. Okay, the human baby, which is they will become you. So like this in the offspring, right? Okay, in every of the, in every of their somatic cell. What is meant by somatic cell? Somatic cell is referred to the body cell. Okay, for example, is the your skin cell, liver cell, bone cell. So that will be the somatic cell. So all of your cell, right, inside the cell, they have about 46 chromosomes. Okay, actually, this is also proven. This is also proven by the scientists. Okay, because they really study the number of chromosomes inside every of the human body cell. Yeah, so they will find out it's 46 chromosomes. Just accept the 6 gametes. It will be only about 23 chromosomes. Yeah. So this is the meiosis. So meiosis is a process they will produce the haploid, the haploid cell. Okay, haploid cell. So we can have a look. Yeah. So how is the process in the meiotic cell divisions? Okay. By this once cycles of the meiotic cell division, right? You can, from one parent cell, you can get about 4 haploid cell. One parent cell, you get about 4 haploid cell. So you need to undergo 2 process, okay, which is meiosis phase 1 and the meiosis phase 2. So you can see, right, you will have first meiotic divisions and second meiotic divisions. So in the first meiotic divisions, right, you are actually involved the pairing and separations of the homologous chromosome. Yeah. So and then what we do is you actually separate the homologous chromosomes. Then in the second meiotic division, right, you are actually separates the sister chromatic for each chromosomes. Let me show you. So you can see, right, this is also same to the mitosis just now, which is before you just start your meiotic cell division, right, or the meiosis, before you start your meiosis phase, right, you need to duplicate your DNA also. You need to replicate your DNA, okay? Like this, okay? Make the DNA into 2 copies. So now each of the chromosome, right, they will have their sister chromatic. okay, they will have a sister chromatic like that, like this. Yeah. And then you have about a pair of homologous chromosomes. Okay. So at this point, what is meant by homologous chromosomes? Homologous chromosomes it refer to one chromosome is from your father's and another one chromosome is from your mother's. So this calls what? Homologous chromosomes. And then they will always in the pairs, okay, a pairs with the same length. You see the same length, right? Just same in their length and the same locations of the centromere, okay, same location of centromere, and then there will be the same gene locus. Yeah, so this calls homologous chromosomes. So we can see, right? Yeah, in the prophase one, okay, prophase. Oh, they are actually involved in the pairing up of the homologous chromosomes. Okay, let me show you another screen. Because I need to draw. Sorry. Okay, I need to draw. Okay, so you can see, right? Oh, yeah. This is the in the meiosis. Okay, this is in the meiosis 1, which is the first phase of the meiosis. So what they do is they are actually involved in the pairing up. The homologous, they just pair together. Pairing up of the homologous chromosomes. What is meant by pairing up? Pairing up is just like this. Okay, you can see, right? This is the homologous chromosomes, okay, which is one from your father's, one from your mother's. So when pairing up, right? So for example, like this is your father chromosomes, okay? And then this is your mother chromosomes. Pairing up just like this, which is their sister chromatic, right? They just contact each other, like this, okay? So what will happen is, and then this is very important, is because this is we call as what? We are call this process about the sister chromatic just contact each other, we call it as the crossing over. Crossing over. Due to the pairing up of the homologous chromosome, right, you will have the crossing over. So what is meant by crossing over? The crossing over is referred to the this one. Okay, this one. So like this one is crossing over. Crossing over. So what will happen during crossing over? There will be the exchange of the genetic materials between your father chromosomes and your mother chromosome. So at the end, right, your father chromosome, they just get some part of their mother chromosome like this, okay? And then your mother chromosome, they just get some part of your father chromosome. Like this. So this crossing over is very important because it is a method to increase the genetic variations. So you know how meiosis can increase the genetic variation or not due to the crossing over. Because this crossing over, they can do what? They can allow the exchange, okay? Exchange of the genetic material, okay? Genetic material. Between the maternals and the paternals, okay, chromosomes. So they can produce different genetic recombinations. A new genetic recombinations of the chromosomes. So it will help to do what? It will help to increase the genetic variations. So that's why we always talk about meiosis, right? The advantage of the meiosis is because you are able to increase the genetic variations. Yeah, like this. So genetic variations. So after they just do the crossing over, right, which is in the prophase 1, they will align themselves along the equator. Okay, so you can see, right, this is the equator. Equator means that the chromosomes, right, they just align themselves in the middle, we call it in the middle phase, right? So you can see here, like this. You can see a straight line, right? Just something like this. So this is called metaphase, which is the chromosomes, they just align at the middle line. So with the spindle fibers attached to the chromosomes. Yeah. So later, during the anaphase, spindle fiber, they will just contract. So when they contract, they will pull. Now they will start to separate. So you can see, right? And those are, they are actually separate the homologous chromosomes to the opposite pole. So go to here, another one go to here. This requires what? Opposite pole. So they are actually separate the homologous chromosomes. Please remember to add this term, homologous chromosomes, because this meiosis phase 1 is the process of separating the homologous chromosome, which is one from your mothers, one from your fathers. And then followed by the division of the cytoplasm, which is a cytokinesis. And then they will enter to telophase. Telophase, which is now the chromosomes, right? They will uncoil and then they become the chromatin like this. So, okay, by this method, right, which is in the first phase of the meiosis, from one parent cell, okay, one parent cell, which contain the diploid, right? The diploid number of chromosomes, they'll become what? They'll become two haploid cell. Okay, two haploid daughter cell. From one diploid parents become two haploid daughter nuclei. But it won't stop. It will continue. Okay, it will continue. Because this is not the end of the journeys. So now they will continue enter to the second phase. Yeah, to the second phase. So means that in the first phase of the meiosis, after they just complete to separate the homologous chromosomes, right, they can produce two haploid daughter cell with each of them consists of the duplicated chromosomes. Yeah, still have a duplicated chromosomes are formed. Now they will enter the second nuclear divisions, which is the meiosis phase 2. So in this process, right, the purpose of the meiosis phase 2, right, is they just want to separate the sister chromatic. So initially it's just like this, okay? Yeah, which is the chromosomes. Each of the cell, they have a haploid number of the chromosomes. Because only one set of chromosomes, so requires what? Haploid number of chromosomes. Yeah, so they break down the nucleus membrane again, like in this way, okay, see? And then they will enter to the metaphase. So during the metaphase, right, they also align the chromosomes at the equator with the spindle fibers attached to the chromosomes like this. And then in the anaphase 2, what they do is spindle fibers, they will contract. They contract. So when they contract, right, they just pull the sister chromatids. So now in the meiosis phase 2, we talk about separate the sister chromatids. So you know what's the difference between the homologous chromosomes and the sister chromatids? Because in this chromosome, right, yeah, you actually separate this. Yeah, so this is actually because what? Sister chromatid. Okay, sister chromatid. So at this one, what is meaning of the sister chromatid is referred to the chromosomes have another duplicated form, which is the chromosome have another copy, same copy. So that's why they are just attached to each other. So this we call it sister chromatid. So they actually separate the sister chromatid like this. So after the separations, sister chromatids, now they will transform, become chromosomes. Now they are called as chromosomes after they just finish separations. And then they will get into the telophase, right? Telophase, which is they will just reform the nuclear membranes. Yeah. And then the chromosomes, they will uncoil, become chromatin again. Like this. Okay, so by this way, you can produce about 4 haploid daughter cell. 4 haploid daughter cell, which is each of the cell contain about 1 set. So you know that there's about 1 set of the chromosomes. It's not 2 sets, but it's 1 set of chromosomes, right? So you can see 1 set of chromosome. If compared to the mitosis, right, we have about 2 sets. Yeah, so you can see, yeah. This is the mitosis. You have about 2 sets. Like this is 2 sets. Like this, right? Is 2 sets. Diploid, 2 sets. Okay? If there's only like this, is 1 set. Yeah, it's because haploid. So at this now you can now you can differentiate. Yeah, you can differentiate for the mitosis and the meiosis. Yeah. We go out. Okay, so so you can see this is in the mitosis, right? In the mitosis, okay, each of the chromosomes they have about 2 sets, like this, 2 sets, okay. But in the meiosis, each of chromosome they only have about 1 set, just one single set on. Yeah, like this, okay, 1 set, 1 set, yeah, 1 set. So that's the difference. So because in the mitosis, right, in the mitosis, you are actually producing the diploid parent cell to get the diploid daughter cell. But in the meiosis, right, you're actually getting the haploid daughter cell from the diploid parent cell because they only have about 1 set. Yeah, only 1 set of chromosomes. Okay, so that's all about the meiosis. So just want to make you become more clear for the process. Okay, so we talk about the meiosis and the mitosis. We already finished talking about this one. And then now we want to discuss about what? Okay, now I think we can discuss some of the questions, right? Yeah, we can discuss some of the questions. Okay, so for this one, what is genes? Okay. And then the exam, they like to ask this kind of questions. They will ask you to define, okay, give the descriptions for the genes or the mutations. When we talk about genes, right, the genes is actually we refer to what? A length of DNA. What's mean by this? Because, right, the DNA is just very large and very complex. So maybe you can just imagine like this. This is a DNA. DNA have about 2 strands, right? It's double stranded. And then there will be another strand, okay? No ending because DNA is just super long. So you can say this is the DNA, and then where's the genes? The genes is referred to the regions, a short regions on the DNA. For example, like this. So this is just a short regions on the DNA. So if your cell want to produce the proteins later, what would they do is they will just uncoil. They will just uncoil this region. Maybe they just uncoil this region. Yeah, okay. So they will expose to the base, okay? Because the DNA, right, they will have the base, all right? They will have the base like this, okay? Which is the what? The ATCG, yeah, they will just expose their base. So there will be the enzyme come out. So the enzyme can make about what? They can start to synthesize about the messenger RNA. So there will be a short strand, a single short strand of the messenger RNA being produced. So what the messenger RNA do? They just copy the genes from the, it's not copy the genes, they just copy the nucleotide base sequence. So here will be the RNA, okay? So what they do is they just form a, we call it what? Complementary base pairing between the DNA and the RNA, which is the A to U, okay? C to G, and then there will be the T, okay? T to A, G to C. So this is the we call it a nucleotide base. So this is the nucleotide on the DNA, okay? And then this is the nucleotides based on the RNA. RNA. So they will form what? They will form the complementary base pairing between this DNA and RNA. So the RNA, they just like a copycat. Okay, they just copy the information from the DNA, like in this sequence, which is this nucleotide base sequence for the RNA. And then they will just bring out, this genetic information to outside. So that's why the RNA, they will diffuse to outside. From the nucleus, they will just get into the cytoplasm. So once the RNA, okay, which is this short strand of RNA, right? When they get into the cytoplasm, they will bind to where? They will bind to the ribosome. Yeah, they will just bind to the ribosome. So here is the ribosome, okay, just like in this. They have a large subunit at outside, and then a small subunit at the below. Yeah, so this is ribosome. So ribosome, they just read along. So I just put R ribosome. So ribosome, they just read along on the genetic informations on the RNA, just read along, and then they start to form the base pairing. Yeah, okay. There will be the base pairing between the RNA, okay, the RNA, and the requires another with tRNA. Yeah. So actually, this is a messenger RNA. I just put mRNA. Okay, this mRNA. So this mRNA, they will bind to the ribosome. mRNA, okay? mRNA. They'll bind to the ribosome. So ribosome, they can just read along the sequence. And then ribosome, what they will do is, what is the function of ribosome? Do you still remember the function of ribosome? What they do? They do about the protein synthesis, right? So remember? They just produce a proteins. They'll produce a proteins. So ribosomes, they produce the proteins. How they can produce the proteins? So it means that the ribosomes, they will help to join the amino acid together. So just imagine, right? This is the amino acids, okay? This is amino acids. So there will be the bond, okay? The bond holding the amino acids together like this. So like this, right? So the bond holding the amino acids is the what? What is a bond? It's a peptide bond, okay? I hope you still remember the peptide bond. This one, peptide bond. Yeah. Peptide bond. So that's why ribosomes help to produce the proteins by forming this polypeptide change. So later, this polypeptide change, right, they can coil to each other, fold to each other, and then they'll become the structures, actual proteins and actual protein structures with different shape, yeah, with different in their proteins shape. So this is we talk about how we're able to produce the proteins. So this is referred to like, okay, this is a DNA, okay, this is DNA. So DNA is just very long, very complex, right? So at the DNA, they will have different regions. So for example, like these regions, right, they contain gene A. Gene A. So gene A later, they can produce about the, for example, protein A, okay, protein A. But there will be another, this location of the gene. They have about gene B, okay, gene B. Yeah, so this gene B, they will produce what? Protein B. Yeah, you can just imagine that this gene A and gene B, they have different functions. For example, maybe this gene A, right, they're just responsible for the hair color, but the gene B, they're just responsible for the for the pupil color. Yeah, so they something like this at the same DNA, right, but different length. It's not different length, sorry, different regions on the same DNA. Same DNA strand, there will be the different region. So these different regions, they will be responsible for different type of the proteins productions. So that's why the genes, what I mean by genes, gene is a length of DNA, which is a short segment of the DNA. They can code for the proteins, which is the nucleotide base sequence, right? The nucleotide base sequence found inside the DNA later, they can provide the instructions to make the proteins later. Because the one who making the protein is a ribosome. The one who making the protein is a ribosome. And then who gives them instructions? It's a DNA. Yeah, DNA, they will provide the guidelines and the instructions to the ribosome to make the proteins. Then what's mean by mutations? Okay, mutations, right, is usually, actually there will be two types of mutations. And that's one. you will be learning two type of mutations, but you just need to know gene mutation first, okay? What's meant by gene mutations? It's because that if you have like any change in the nucleotide base sequence, for example, I show you like this, okay? Sickle cell anemia, you know? Sickle cell anemia is a type of the gene mutations. Yeah. Let me show you. Where is the sickle cell anemia? So you can see, right, this is a sickle cell anemia. So how they can produce this sickle cell anemia is due to there's only one base undergo mutations. So sickle cell anemia is the gene mutations. So you can see, right, single point mutation on this gene, which is like this. Okay, they just change about one base sequence. Just that one base sequence, they can cause the proteins become abnormal. So like in this normal people's, the DNA is like this sequence, okay, GAG. But in the sickle cell anemia, right, the GAG, the A, they just mutate, become T. So when they become GTG, right, they will produce different form of proteins. Yeah. Okay, because this this nucleotide base sequence, they can undergo the translation later, and then they will become the proteins. So that's why they can allow the amino acids connect to them. But in this sickle cell anemia, due to that once base being changed, so they will have different proteins, which is the valine instead of these glutamines. So that's why these mutant proteins, right, they will cause the red blood cell, right, they just appear like this. So you can see, right, sickle cell anemia. Sickle cell is referred to what? It refers to the red blood cell, just like a sickle shape, you know, sickle shape. So this sickle cell anemia or sickle cell disease, it will cause what? It will cause anemia. Anemia, which is the patients will be lacking out of the, it will reduce. Actually, what they do is they will reduce the oxygen carrying capacity of the red blood cell. Because the sickle cell anemia, the red blood cell is just so thin, so fragile. It's sickle shape. Okay, because in the normal, in the normal red blood cell, what is the shape? In the normal red blood cell, right, the shape is, we call it what? Biconcave, this shape, right? In the normal red blood cell, we call it what? Biconcave, this shape. So this biconcave, this shape, and is a very, they can help to increase the oxygen carrying capacity. But if the red blood cell proteins being mutated, they'll become the sickle cell shape like this. Okay, and then this show you about what? Sickle cell anemia actually is a type of the autosomal recessive genetic disorder. Okay, because of what? Because that is referred to the autosomal disease. What's meant by autosomal? Autosomal is referred to your somatic body cell. Okay, it's referred to somatic body cell. They just undergo mutations. So we call as autosomal. And the recessive, it refers to the, you have a dominant genes, you have the recessive genes. Usually the dominant genes, they just refer to they are just referred to the genes, which is they can express themselves. Then recessive genes, they just refer to the genes, right? They can't, they have a very, very weak expression on themselves. So they can be easily suppressed by other genes. So sickle cell anemia, they will only appear if the patient, right, they just inherits two of them, which is the recessive alleles. They just inherit the two recessive allele from the parents. So they'll become the sickle cell anemia because both also is a small a like this, okay, small a. So if in a normal patient like us, okay, it's in a normal healthy persons with the biconcave dish shape, right? So this is our genotype, big A, big A. If the carriers is one big A, one small a. Yeah, so this is called a carrier. Okay. So and then the exam they would like to ask about this one, okay? You have about this one, big A, one big A, one small a, and then you have about two small a. So which one is resistant to the malaria? So the one who resistant malaria is the one who contain the sickle cell gene. Which is this one? Small a, two of them are small a, or only one of them are small a, which is they contain the sickle cell gene. It's because that the malaria, okay, I'm not sure you heard about malaria before or not. And that's young. It's a kind of disease, mosquito-borne disease. The mosquito, right, when they suck your blood, they can inject the parasite into your red blood cell. So the parasite, they can just grow in your red blood cell. So malaria, they just prefer the biconcave dish-shaped red blood cell, like in this one. Okay, malaria, they like this. I mean the parasite, they just like this. It's because that there will be a lot of space for them to do the replications. So that's why the normal patients, even though they don't have the sickle cell anemia, but they will still be susceptible to the malaria. Because the red blood is just very round, okay, very healthy. So just a very favorable environment for the malaria parasite to just replicate inside, as compared to the sickle cell shaped red blood cell, which is they don't have much space for the parasite to replicate inside the red blood cell. Okay, and then for this one, right, genetic question. Okay, this is the exam they will not ask you about this one, but I just want to show you about when you do about this genetic question, right? When you do about this one, yeah, see, when you do about this one, right, because there's a lot of questions just asking about this. Okay, so yeah, genetics was asked to investigate the inheritance of achalatasia in the dogs. And then the normal allele, they will tell you the normal alleles is being represented by the B, big capital B. And the mutant alleles is represented by the small capital b. So what does this mean? One is dominant, one is recessive, right? One is dominant, one is recessive alleles. And then, yeah, they will give you this one, pedigree analysis. It's not pedigree analysis, it's called pedigree tree, yeah. And then you need to write down the genotype, okay? So this one, I'll just show the answer, okay? For example, like you can see, this is this one is normal male, yeah. If they contain the black color, right, is the affected patients, either it's male or female. So the male will be the rectangular in shape. It's not rectangle, it's square. Will be in square box. If in the female, right, will be the circle shape. So how you can guess is like they're asking you about 1, 2, 3, right? The genotype for the 1, 2, 3, which is this one. 1, 2, and the 3. So you should write about genotype. So how you can guess the genotype? So starting from here, okay, this number 2. Oh. Okay, you know that this number 2, right? It is what? It is a affected male. Okay, it is a affected male. And then this affected male, right? Okay, this must be, they must be what? They must be 2 small b. Yeah, 2 small b. Because when you can see this 2 and the 3, right, which is this affected male, if they marry with the normal female, they'll produce a children's, which is the female. And then that female is affected. So that's why you can have a guess. Yeah. You can have different kind of genetic recombinations, okay? You can just have a guess. Okay, maybe the male, yeah, I know the male is either it's like this, right? Or like this. This is for the male part, okay? This is for the male. So if I want to have a guess, how is the genetic recombinations you can produce this? Means is you need to do a lot of the trial, okay? So you can see, right? Maybe I can choose about this. For example, if I choose about this, right? And then I mate with the female. The female, okay, I'm not sure is either is this or is this. Okay, for example, if I try to choose this one, okay, and mating with the but the result that you produce, right? So you can see this is a male part, this is a female. So that's why the offspring, all of them will be the normal ones. You can see all of them will be the normal offsprings. So only about 2 of them will be the carrier. So this is not, this is not following the answer, because the answer showing that female is 2 of them are small b. Okay, because you know that female is affected. So that's why you just delete this. Okay, you know it's about the 2 small b in the male. Okay, what happens if you have 2 small b in the male? So that means you need to produce a 2 small b, okay, at the end. So that's why 2 small b in the male, right? So you can see this is a male. So when they cross with the female, right, they will produce a, for example, this female, okay, they must be the carrier. Why the female must be carrier is because that if the female is not carrier, right, it's also 2 big B, you only will produce all of them are carrier. None of them will be affected. So that's why you should know that either 1B, right, is referred to the recessive. So when you do the crossing, right, it will be like this, okay? BB, BB, and BB. So you produce about two offsprings. Okay, these 2 offspring, right, it is affected. So that's why you produce 1 offspring is 1 big B, small b, another offspring is 2 also small b. So it will be affected. So this is a way on how you're able to find out the answer. Then same to the number 1, okay, number 1. So number 1, finally, you get about the offspring, which is also small b, 2 small b, right? So how you can get about 2 small b? So that means it's either one of the parents, they must be the carrier. Okay, so they will have this one, okay? So this carrier mixed with carrier, right? You'll be like this. Okay, BB. And then BB again, which is 1 big B, 1 small b, and then you become 2 small b. So these 2 small b, they will become like this here. So that's why the answer is 1 small b, 2 big B. Oh, sorry, 1 big B, 1 small b. So you just have a guess, okay? Have a guess on the answer. And then for this one, now you need to draw the genetic diagrams. Okay, so how you can draw genetic diagrams? Let me show you some of the questions they will be asking is, okay, just like this, see? Genetic diagrams. Genetic diagram, right? Actually, they are just drawing about this, which is you need to write out the phenotype. The phenotype is the descriptions. Either it's tall, or it's bright in color, or in dark in color. So this is the phenotype. Phenotype is you use the words. You are using the word to explain them. And then genotype is the alleles, the alleles, the dominant alleles or the recessive alleles. So for example, you want to meet a tall to a dwarf plant, the genotype is about 2 big T because it's tall. And then you make with dwarf plant. So the dwarf plant will be about, still left 10 minutes. Sorry, later I just open new link again. So the 2 small t, right, is belong to the recessive allele. So that's why the plant just appear in the dwarf. Means the plant is just very short. So this is gametes, okay? This is gametes. And then this is your offsprings. So same to this one. Okay? So as well, just remember that if you want to write out your genotype, if you want to write down the genotype is always represented by 2 letters, always 2 letters. There will not be only 1 letter or more than 2 letters represent about genotype. Because what I mean by this is genotype, they contain about 2 letters is because they contain 2 alleles. One is either dominant allele or recessive alleles, right? Because one of the allele will be passing from the mothers. Another one allele will be passing from the father to you. So that's why every of your cell, right, you have about 2 genotypes. Okay, anything I want to show you? Okay, so you can see this is the alleles. Define the term of alleles. So this is also being asking in the exam. How you can write down the terms. Alleles is, which is the one? The big capital A, the small capital A, the one we call alleles. So one genotype, they have about 2 alleles. So alleles it refers to the alternate version of genes, which is the genes just carry same function, but they just appear in a different way. For example, the alleles, right, you have about the hair colors, okay, the hair colors. So these alleles, okay, they're just responsible for the same, which is the hair color, but they are different in color. They are different in the physical expressions, but their function is the same, which is they're just responsible. They're just responsible for the hair color. So this is called as alleles. Alleles, which is refer to the same genes. Same genes carry that same functions, but the gene just appear in different form, different physical form, different in color, different in the length, different in the structures. Okay. So how you able to answer these questions? How you able to answer these questions is, you need to write down the genotype. Dot 4 and dot 5. Okay. Genotype for the dot 4 and dot 5 will be this one. Yeah. 4 and 5, right? So you know the 5 is 2 small b. How about the 4? Okay, a normal dots and the accidental dot. Yeah. So the normal dot will be 2. Just follow the letter. Just follow the letter on the both. Normal. Like this, okay? And then if you have acatasia, it will be like this. And then how's your gametes? Oh, sorry, normal. Okay. Okay. Remember that this normal, the normal dots, right? They can be totally normal genotypes, which is both of them. They also have the dominance alleles, or only one of them is the dominance allele, but other of them will be the recessive alleles. Because normals, right, which is the phenotype, when the phenotype just appear in the normal appearance is maybe their genotype is homozygous dominance or the heterozygous dominance. So like in this way, they're actually showing it is the heterozygous dominant, heterozygous, which is one big and another one small. So it's actually a carrier. It's actually a carrier, but these carriers, they do not express themselves. They just look like normal appearance. They do not express themselves. So even though they just carry that genes, the recessive genes, right, they can, for this acatalasia, yeah, for this acatalasia. So this is the gametes, okay? You need to separate the gametes like this, okay? Like this, like this, like this. And then you need to draw the Punnett square, okay? How you can draw the Punnett square? This is number 2. Okay, the Punnett square is just refer to this one. Let me show you the Punnett square. Example of the Punnett square is this. Okay, it'll be like this. Okay. So yeah, this is talk about genetic diagrams. So genetic diagram can be determined by using the genetic diagram called as Punnett square. So this Punnett square, right, is a diagram they just showing a different combinations of the alleles which could be produced that offsprings. So when you talk about dominance alleles, you're always showing the big capital letter. Okay, if the recessive alleles, right, you will always show about lowercase letter. So one is big T, another one is small t. So you need to draw out something like this, okay, which is a Punnett square. Just similar to this one. So let me draw one to you. Okay, so that's why you have about the parents, right? Which is the dog's 4. Okay, the dog 4s, the alleles will be this one. Another, you have about the dog 5, okay? Dog 5. The alleles is about 2 small b. So just draw a square. Just drawing a square. So here is the, so that's why at the end, you have the different genetic combination, which is you have like this. Like this. Because they are actually this cross with this, okay? This cross with this. And then this cross with this. So the genotype will be this one. And then you can put like this, and then this. This is the genotype. So how is about the offspring? So the offspring, right, you can write about, it's a carrier, okay? And acatalasia. Okay. So like this, you get about full marks. So every time after you finish drawing about genetic diagram, right, you should know how to describe them in genotype, and also you should write them in the phenotype. And then state the name that type of cross that you have complete is actually, this is a test cross. What's meant by test cross? What's meant by test cross? Test cross is, maybe you can show this one. So you can see the test cross. Where is it? Test cross. See, this is a test cross. So the test cross, right, is referred to an experimentals, a type of experimental cross of individual organisms of the dominant phenotypes, but unknown genotype. It's because of this dominant phenotype, right? They have about 2 genetic combinations. Either one of them is about one big B, one small b. Either is the 2 of them are also big B. So either it's heterozygous or homozygous. But you are not sure, so that's why you need to do a test cross. You need to cross with the homozygous recessive. So you need to observe the offsprings. So by this way, you can guess the parents' phenotypes. The parent genotypes. It is from the heterozygous or it's from the homozygous dominance. So this is test cross. So at this one, just remember the function of the test cross is they will just provide them some methods. So that's why they can trace back their parents is heterozygous or is a homozygous in their genotype. Because if you make for example, if you make you can see, right, both are also purple flower made to the white flower, but the result is different. Because one of them is heterozygous dominance, another one is homozygous dominance, right? So that's why you can produce a different result. In the test cross ones, which is when you cross this homozygous recessive alleles with the heterozygous alleles, right, you will have about 50% of them is purple flower, and then 50% of them is about white